Okay, so it's a pleasure to have Nataša to give the talk. It's unfortunate that, I mean, the hope was that she would actually come up to Rutgers and we could spend a couple of days talking, but someday in the future that will happen.
Yeah, hopefully.
Hopefully. And she's going to talk about algebraic and topological models for DNA recombination. So you should take over the screen and.
So can you see my slides?
Yes, perfect.
Okay. All right, let me see if I have option to, I'm sorry, I clicked the wrong button. It seems that, let me see if I have option. I don't have an option to have the pointer.
But we can see your cursor.
You can see my
your mouse.
Ah, yeah. Okay, that's good then.
Yeah, it's perfect.
I guess I don't need it. Okay, so it will be mostly a story about how biology and math can interlace. And before I start, I want to say happy birthday to another person that is, I think, present, Laura Landweber, who is my collaborator and with whom I have been working for many years now. And I think she did join the talk. I think at least I saw her on the list. Are you here, Laura?
Yes, thank you.
Okay. Well, happy birthday. So usually when I start speaking in front of a mathematics audience, I want to point out some personal journey. And my personal journey actually started when I was, in some sense, I really wanted to, what, am I here? Okay, yeah. I wanted to learn what is really a gene, because I know that people have been talking about it, but as a mathematician, if you want to speak about something, you want to know a definition of it. And I was very excited when I see this paper. It's already almost 15 years ago. What is a gene? And I thought that now I can learn something. And it turned out that the paper didn't really answer my question. It didn't give me a definition. What the paper showed was that, well, as you can see, the site here, the idea of genes says beads on a DNA string is fast fading. And RNA is a key part of the information package. And right now, if you speak to biologists, they're all really interested in how RNA is involved in, I guess, preserving and reorganizing of genetic material and controlling, for that matter. So this is where my journey, I guess, started here, when I realized that the genes are really, really complicated and I cannot really define it in a precise manner the way a mathematician would expect it to define. And, you know, if you're studying a genome, then you can say, oh, from here to here, that's a gene. But it turns out it's not that simple. So we started studying DNA recombination and gene rearrangements. And I just want to point out that these processes are very common in biology, and they have been observed in many instances, both evolutionary and developmental scale. And what in particular shows up is certain species of ciliates redefine gene processing on their own, and they're used as model organisms to study DNA recombination. So I will give you a prelude of the model that we have been looking at how to study DNA recombination. And so let's imagine that we have a DNA sequence of this sort, and that each one of these colored segments are segments of DNA that have been or would be rearranged in the sequence that is at the bottom. So that means that I have removed certain segments of the DNA. These are not non-colored portions. And also we have rearranged them in some way. The way that we can assume this might happen is when there is small segments. So I think we can imagine these larger segments to be like several hundred nucleotides, but the smaller ones can be, say, shorter, 2 to 20 nucleotides. And maybe this is a good color. So you can see that some segments and with short sequence that is repetitive add another segment, and in some sense can be used as pointers. So this small segment here can point to this other segment, and we use that to recombine these two larger segments together and form this portion of the molecule. So if I assume that this is how the whole situation might be happening, then I can associate symbols to each one of these shorter segments at the end of the larger segments. And these symbols now, if I start reading them throughout the molecule, they form a word. And now I can look at these words and work from here into the recombination process. So once I have these words, imagine what happens with these words is that every symbol appears twice, and we want to call them double occurrence words because the symbols appear twice in them as pointers. So a possible model, we continue with this. If I look at this word, then I can associate a graph to this word. And this graph is such that if I go, every symbol is a vertex. And if I go from one symbol to another, that means I go through the vertex straight. I go straight and then continue and then go straight through the vertex. So the graph has, every vertex is degree 4. I can imagine it is like a cross. I don't know if you see my fingers. And whenever I come to a vertex, I go just straight through the vertex. So if I start reading this graph with this sequence, I can start read A, B, E, F, and then G, C, B, and so forth. And in fact, I can read this segment, this sequence here. Oops, didn't want to do that. From this graph, I can also consider a chord diagram. This is another mathematical object that we can use. A chord diagram are often used in biology when the data is presented. But in this particular case, this chord diagram is also a reflection of the word and the molecule that we describe with this graph here. The chord diagram connects symbol. Well, we arrange the symbol, the word. We arrange the word around the circle, and we connect symbols with the same symbols with the chord. To this chord diagram, of course, we can associate now a circle graph, so which is the intersection graph to this chord diagram, and the circle graphs and other objects that have been studied quite a bit in mathematics. And they appear quite often, in fact, in various. I'll go back now to the model organism, and we have dealt with data from Oxytricha trifalax. And there are several species actually of ciliates that go through some massive recombination processes. And this is where my interest, or our interest as mathematicians, can be, intrigued, I guess. Just a short description of these organisms. They are one-cell organisms. So this here is an image when two ciliates mate, and they have started in exchanging their genetic material. But let me go back and explain how this works. Normal process, normal life of a ciliate actually goes through cell division, and it goes through this, I guess, asexual vegetative growth. But when they're stressed, they go through this other cycle. And this is the cycle that we are interested in. A main feature of these types of ciliates is that they're binuclei. So they have two types of nuclei. One is a micronucleus and the other one is macronucleus. The micronucleus is zygotic nucleus, usually is not responsible for the life of the organism. But the macronucleus is a somatic nucleus where all of the genetic material is, well, all of the, I guess, life processes of the cell are being performed. This is not exactly true, but almost true. In any case, when they're stressed, and I believe Laura in her lab, they starve them, and when they are very in that situation, they start mating and go through the cell conjugation. And this is the part of the cell conjugation. So they join together, two cells, two mother cells join together. They exchange the micronucleus or micronuclei, and also they exchange the genetic material of the micronucleus. At some point, the daughter cells start disintegrating all of the macronucleus. And the macronucleus is, I guess, disintegrated. The gene, the DNA has been, I guess, chopped up. It's not part of the nucleus anymore. And they slowly use the newly developed, newly formed micronucleus to go through a large process of large amplification of the DNA, and to rearrange their genetic material and start building their new macronucleus. And this is the part here. These arrows are the processes when this massive recombination processing happen. And this is where we are interested in learning how these things are being done. Here's an example. This is an actin 1 gene. And this is in the this is how the gene appears in the micronucleus. And this is how the gene appears in the macronucleus. So the micronucleus being a zygotic, being dormant most of the life of the cell, actually somehow is used as a storage material because it keeps the DNA, but the DNA segments that it appear in the micronucleus are all shuffled, and they're not a functioning gene sequences. In order to get the functioning gene sequences, they have to be rearranged and recombined, and they're the way that they're done is in the macronucleus. During this process that I just mentioned, this process of building the new macronucleus out of the old micronucleus, newly formed micronucleus, not old. Okay, so this is what needs to be done. So in the micronucleus, the gene may appear in completely shuffled segments. There are small portions, well, not small, but there are portions in between these segments that are eliminated. Then these numbers here, 5, 6, 7, that appear at the ends, those are the pointers. They're repetitive sequences that in some sense guide the rearrangement. And this happens to thousands and thousands of genes during this process. So just the jargon, because we will be using this jargon. MDS are macronuclear distinct sequences. So those are the chunks like this. ISs are internal eliminated sequences. Those are these black bars that connect the MDSs that need to be eliminated. And the pointer sequence are the segments of the DNA at the end of one of the MDSs, MDSI, and beginning of MDSI plus one. They're repetitive sequences that flank the MDSs, in some sense guide the recombination. It is assumed that the pointer sequences appear in pairs, like 5 here and 5 here, and only in a single copy in the macronucleus, in the macronuclear gene segment. All right, so how one can explain the actual process and how do they do this? I'm going to talk about one approach, which is the intramolecular model, which I just described. And here the people that we have originally we started working in. This is a different picture than Laura has now in Zoom, I think. But Laura has been our collaborator for a long time, Masahiko Saito, and Angela is our former student. So we proposed a model that is sort of suggests an explanation of this recombination process through RNA template. And this is why I started my talk with having RNA involved in gene processing, because being part of the whole genetic, oh, okay, something happened. So we propose that there is an RNA involvement and there is an RNA template that guides the recombination process. So here's a cartoon picture of that. So imagine that I have two micronucleus. So this is micronuclear DNA, and this is another micronuclear DNA. I have MDSI. So the ith segment of the MDS is here, and the I plus first segment is here. Then these are ISs that need to be extracted. And I have RNA template, a molecule that is complementary to the portion of one of the MDS, some part of the MDSI plus one and some part of the MDSI. If this RNA actually goes through branch migration and makes this R loop or invades the DNA, the segment of the DNA that is not attached to its, I guess, sister DNA becomes free to interact with the other segment of the DNA on the other side. So this is guided through the pointer sequence, which is, as we mentioned, the same in both cases, and they're complementary to each other. So with this cartoon, we propose that this RNA can actually guide the recombination process. If I think about this happening around the pointers and the MDSs, I can describe this as a vertex in the 4 regular graph, in the graph, as I pointed out at the beginning. And by doing the recombination, then I think about the smoothing of the vertex and joining the ends. Now, this graph of actin 1 that we just showed in the beginning corresponds to this word. And this is how we can describe now the whole situation. This picture here is described here because if I go through the graph straight through the vertex, I read the micronuclear gene. But if I, at every point, I turn to the right immediately, then we can describe the macronuclear gene. So if I start from here and go to the right, everywhere I end up with the macronuclear gene. And when we smooth at the vertices, so this is my graph, and if I smooth the vertices according to where my MDSs show up, we end up precisely with the order that is necessary for the gene to become functioning gene. So we have 1, 2, 3, 4, up to 9 in the given order. So mathematically, we can describe this, we can propose to study these assembly graphs, which are now graphs with rigid vertices all have valency 4 or 1. We can talk about the transversal, which is the component that goes through the vertex straight. And we want to study simple assembly graphs that contain only one transversal, because if we talk about only one molecule interaction or one mic molecule, then this will be a simple assembly graph. So here's a picture of 2 component graphs. So I have a blue and a red. The red is cyclic and the blue is not. And if I just have one component, then one component will be a simple assembly graph. And this is the types of graphs that we will be looking at. So here's a picture of small size graphs. These are graphs, and in fact, actually all of them show up in the data. Oh, I forgot to mention that once we write down the word, we get the double occurrence word. So the word that is formed out of the pointers, we end up with a double occurrence word. And this double occurrence word, as we know, is also known as a Gauss code in many instances. So we have pointers that are letters or symbols in the Gauss code. We can label the crossing or the vertex with positive, P, if both appearances of the pointers are in the same orientation, and P bar if one pointer is in the reverse orientation than the other. And we perform the smoothing. So we have a positive smoothing if we have a positive labeling on the crossing, and a negative smoothing if we have a negative label on the crossing. So for simple assembly graphs, we can actually show that if for every sequence of smoothings that we can have assigned to our vertices, positive or negative, we can embed this graph in the space such that after performance of this smoothing, all of the components are unlinked. So the molecule is not going to be knotted or linked. Another observation. Okay, so let me go back to the theorem that we can state here. If I take a micronuclear arrangement, so this G, which is the genetic sequence, I can think of a double occurrence word W sub J. K sub J is the graph that I obtained from the double occurrence word. M sub I are the MDS segments, and I sub J are the IES segments. So both of these are labels of the arcs. So we label the arcs on our graph with MDSs and IESs. So this labeling is alternate. They alternate M and I's. And it is very similar to the Kaufman proper coloring labeling on projections of virtual knot diagrams. But it turns out that the smoothing is not the same. And when we perform the smoothing with positive negative smoothing, we end up with different components and different types of components and links, actually. We can prove now, if I have this setup and I form all possible graphs for any types of rearrangements that we can see in the graph, after the smoothing of the vertices, there will be one component that contains all of the MDSs in order 1, 2, 3 up to K. And this actually explains how the molecule actually can recombine into a proper order in the macronucleus to get to the macronucleus. Before I go further, well, everything that we wanted to talk about here is about spatial graphs. So the graphs are in space. They're I think my pictures here maybe have indicated that, for example, this graph here is not a planar, and we cannot sketch it as a planar. And there's an interesting graph that showed up in the studies, and this is called the tangle chord. So how we build up this tangle chord, I start with the word 1, 2, 1, 2, and then add 3 at the end, but with the last one in between, last vertex in between. Then I add the next symbol with the last one in between, and so forth. By doing this, we end up with a graph of this sort. So the tangle chord is kind of interesting. It shows up later on. But we started looking at genus ranges of these graphs and wanted to see how these graphs, if indeed they happen, of course, there's at this point no evidence whatsoever biological that these graphs do appear during the recombination. But mathematically, it was interesting to us to study how these graphs can be embedded in space, because, well, we know that they can be embedded so that once we smooth everything, they are not linked or knotted. But let's figure out how they can be embedded and what is the genus range of these graphs. So genus range of graph will be the values of genera over all orientable surfaces of F, into which K is embedded cellularly. So we need the cellular embedding because I don't want to add additional handles in my.
Okay, bye.
Yep.
Oh, sorry about that.
There's a question?
No, sorry, my wife said something and I was just responding. I'm
sorry. Okay.
Actually, Natasha, sorry, there is a question now that there's been a, what is genera?
It's a plural of genus. Genus of the sequence, genus of surface, right? Where we embed graphs.
All right, got it. Thank you.
I mean, a graph can be embedded in many different surfaces, right? With different genera. And the range of the genera, right? The values of the genera of these surfaces will be my genus range. But I want cellular embedding so that there's no attachment of arbitrary handles at the end. So it has to be, the complement must be cells.
Thank you.
So there are questions that we were considering how this, these are in fact the questions, what sets of genus ranges can actually appear for these graphs. And we want to identify the class of 4 regular rigid vertex graph with the given genus range, right? Given the range, what types of graphs actually have this range? So in order to do that, we started actually, you can thicken the graph and look at the ribbon graph as being a ribbon or two-dimensional manifold with boundaries. So every edge now appears as a ribbon. And when I talk about, it turns out that this question mathematicians have not looked at, although it's a kind of natural question. In particular, when you have rigid vertices. So if I'm fixed on rigid vertices, then looking at the genus range for these graphs is turned out to be open. So there are 2 possibilities to connect. Once I have this connection, I can connect this edge like this, or I can connect this edge from this side. And if I look at the boundary components around the vertex, they're either this way to consider them, or I can flip the vertex by itself, one side, and this is another connection, or this other connection can be described by this flip here. And as we know, the number of boundary components actually is important in figuring out the genus of the surface where the graph is embedded. And this is an old formula. M is the number of vertices, B of F is the number of boundary components for the ribbon graph. and this is the genus. So it's an easy formula to compute once you figure out the boundary components of the surface. When you investigate all possibilities, you see that by changing, there are only two possibilities for every vertex. By changing the connection of the boundary components at the vertex, these are the possible changes that we have. Unfortunately, this is not very, they're tiny. I hope you can see them. But there, I can either the number of boundary components are stay the same, or they change by 2. And 1 goes to 3 or 4 goes to 2, that's it. And this is how you can look at the tangle chord. So this is a tangle chord with 3 vertices. These are all 8 possibilities for every vertex. And they're actually complementary. And they're the same. If I take one type of connection at every vertex here, and I flip all of them, then the number of boundary components doesn't change. Here I have one type of connection at these 2 vertices and another one in here. If I flip them, then I have the same boundary components and so forth. But these are all possibilities, and you can see for this tangle chord with 1 and with 3 vertices, I have either 3 boundary components or 1. So another thing that we looked at, because the assembly graphs that we are considering have these loops that pop up all the time. What happens if we have a loop addition to the graph? That means if I have a graph that corresponds to word W, I can add a symbol AA at the end, and that adds a loop to the graph. Well, it turns out that the genus range doesn't change. So loop addition doesn't do anything. And loop nesting, which is adding loops one after the time, also doesn't change. And in fact, we can show that the word W is loop nested if and only if the genus range is just 0. It's not, it's a planar graph. What happens if you add a pretzel to an edge? So imagine I have an edge here and cut the edge and add the pretzel to it. Well, if my graph had the genus range AB, interval AB, then I end up with either interval AB plus 1, or I add 1 to A as well. So I add 1 to the genus range. And there are other observations. The one observation is that for any positive integer n, there is no graph of 2n minus 1 vertices of genus 1. So that can appear in only singleton genus n, which is the highest genus, in fact, that you can have if I have 2n minus 1 vertices, and can be embedded only in surfaces of genus n. However, there are graphs that attain the full range between 0 and n, all possibilities. Oops, okay, something. So if I order the intervals, and the intervals are ordered like this, 0, 0 is smallest, then 0, 1, then 1, 1, and so forth. If I order the intervals like this and I list them at the bottom, this is the distribution of the graphs and with respect to the genus ranges. So this is the order of the intervals from 0 to 3, 4. For graphs of 7 vertices and for graphs of 8 vertices. As you can see, they're quite distinct, and there's distinction between odd and even number of vertices. But there are graphs with singletons like 3. So there are graphs that can appear only they can be embedded only in surfaces of genus 3 and nothing else. But however, we could not find something at the highest genus. Okay, where am I? Oh, here. So the main observation turns out that these are the intervals that we can see. All intervals can be actually achieved by these assembly graphs. If I have odd number of vertices, then there's these dots here are empty. That means those graphs don't exist. And if I have odd number of vertices, I have nothing on the diagonal. We can prove that, but we have no idea whether that happens also when we have even number of vertices. We believe that there might be a bound after which everything, well, to be honest, I don't know. Maybe there are gaps here, or maybe this line can be populated, but at this point we have no idea what's going on the diagonal. But all of these other ranges can be achieved with distribution quite unusual, as you can see. The types of graphs, the size of graphs, no, the number of graphs that achieve certain interval of genus ranges. Okay, I'm going to go back to the data now and reference a little bit more about what's showing up in the data. So in Laura's lab actually reported the whole genome sequencing of Oxytricha trifalax, and this is a snapshot of what has been considered. There are about 16,000 macronuclear contacts, and every contact you can think of it being small chromosome, because if they have telomeric sequences on the side, because these chromosomes in the macronucleus are 1 through 8, from 1 to 8 genes on the chromosomes. So they're small chromosomes, but on the other side we have about 16,000 of them. And they all appear, and Laura can probably comment on that, but I think that they may be between 100 to 1,000 copies of each chromosome. So once we have the sequences, we wanted to take a look at the arrangements of this micronuclear versus macronuclear segments, and what types of patterns appear and what types of graphs we can detect in the data. First, we wanted to observe that during the sequencing, you have no control which DNA you have been sequencing. And sometimes for some genes, it may be sequenced the one part of the DNA, but the other one, for some other gene, maybe some other DNA. If we are looking for patterns, then you can have different rearrangements that correspond to the same molecular arrangement. So if I read 1 and 3, so I have flagged this light blue segment here. This light blue segment appears as fifth, 1, 2, 3, 4, 5 in here. So if I read 1 and 3, in 1, this fifth segment is actually in the reversed order and appears at the end of the mic. If I read 2 and 3, if I read from here this way and 3, then 2, this blue segment is in the right order, but appears as a first in my sequence. If I read 2 and 4, so if I read 2 and 4, then this light blue segment is now 4, but it appears first in reversed order. So it's now M4 rather than M5. So all of these arrangements are actually making one equivalence class for the type of arrangements that can be seen as in the data. And we made a convention which representative of the equivalence class to take. So we decided to take minimal inversion, minimal number of inversions, smallest lexicographic order of the MDS order, and we wanted to delay the inversions as much as possible. Oops. So this is a table of types of arrangements that we observed. The most prevalent ones are these even and odd sequences of MDSs. So you have odd followed by even, or even followed by odd. And as you can see, all of these guys that we have identified earlier are also odd even, and these are also odd even. The only outlier on this in this list of odd even is actually this one here. And there's some complex stuff which we cannot be listed in a simple table. So there's some evidence that this IESs in between two consecutive MDSs, which they're called, or we call them conventional IESs, are removed early in the process of recombination. So what we did, we just eliminated those IESs, and those IESs correspond to a loop like this, 5, 5. So in some sense, I can eliminate this IES and I glue M5 and M6 together to have one M5. And I reduce 5, 5 from my double occurrence word also when I wanted to consider the patterns. And another thing that we also did was we first removed the loops and then we reordered the pointers, relisted the pointers in, this is the remaining pointers word, in ascending order. So the first symbol appears first, the second symbol appears number 2, the third symbol is 3, and so forth. So once we do this, the isomorphism class of the graphs is the same, but we have a unique way to represent these classes now with words written in ascending order. So most prevalent were these situations in the data, but the other ones of even odd situations turned out to be to correspond segments in the word that are repeat segments, 1, 2, 3 up to n, and then something, and 1, 2, 3 up to n, and return segments 1, 2, 3, n, and then n going backwards 3 to 1. In both cases, we have removed the loops. Now we looked at the pattern. So this is a precise definition of a pattern, what we mean by a pattern. But the interesting patterns, the pattern would be a word that is a word of variables. So these alpha 1 up to alpha n are variables. And I can say that this pattern P appears in W if I can map each of the variable to a subword in the word, so that the same variables map into the same subword. Here's an example. There are only 2 patterns that I want to talk about. So one is a repeat pattern, which is alpha alpha. So that means there's 2 segments in my word that appear together. And the other one is return pattern when we have alpha and alpha reverse. So I have A1 up to AK and then AK back one to A1. So these are the 2 patterns which correspond to my repeat and return words in my in the data. So this is another snapshot of the graphs that can be associated to the arrangements in the data. The most prevalent graph, well, this is only for 3 vertices, graph with 3 vertices. The most prevalent one is repeat 1, 2, 3, 1, 2, 3, and graph of this sort. We also looked at the nested appearances of these repeat return words. So imagine that I have a graph like this. So nested means that I have an instance here, 1 and 2 of repeat pattern. We remove this repeat pattern. Then I look at the word that remains. I find in the word that remains another pattern appearance in here. I remove that one and look at the next word that showed up and so forth. So this can be described with, imagine that I have a graph of this sort, and these are all repeat or return patterns. And if I remove them, smooth them out completely, pull this back on this side, pull this end back on this side, I get with another one, with another pattern of the same sort. So these are nested appearances of repeated and return words. So here's an actual data contig 19801. Okay. It corresponds to graph like this, and there are repeat return patterns here. These are the boxed portions. So I start with this graph. We remove the loops, and then we look at the repeat and return patterns. We remove those. And what end up is another repeat return pattern. As you can see, I have this one here, 1414, that shows again. So these are nested, 2 instances of nested repeat return patterns. It turned out that the data was full of these nested repeat return patterns. So we did this iteration of removing of repeat return patterns at every step. And about after 4 iterations, some of the graphs stopped. That means there were no repeat return patterns remaining to be removed. So there were about 176 of those that cannot be reduced and further. And out of those 176, they were about 28 were very complex. That means that there are no repeat return patterns. That means no instance of iteration can reduce them. The other one, about 123 stabilized after 1 iteration, 1 removal of repeat return words, and the rest of it is some complicated stuff, and so forth. So what happens to those that we cannot reduce? So that means that these patterns of repeat return words cannot be detected. Not even nested sequences of them can be detected. So let's go back to the chord diagrams. And this is how repeat pattern shows up. It's a click, and return pattern is actually set of isolated vertices. So those are the 2 extremes. Here's a contig 582 that looks like this. So I have repeat and return patterns in green and blue, but I also have these other chords that have nothing to do with repeat and return patterns. And there's some other more complicated stuff. Again, here you have repeat and return patterns, but the other ones are, yeah, not there, or something like this. And remember that there were lots of loops removed. So these contigs actually have hundreds of vertices. Well, we detected some level of presence of this tangle chord that I mentioned earlier. And the tangle chord, well, did I say something about the genus? I'll mention that later. So this tangle chord graph of this sort is somehow embedded in these guys here. So, well, these are the 4 main situations that we can say that appear most prevalently in the data. What is the magic behind that? Well, we don't know. We certainly know that repeat and return patterns are not our invention. They have been observed way back since the beginning of the studies. But of course, there was reporting of 1 gene or 2 genes that have these. Our study was the first one to say, well, in the whole genome, you have a prevalence of these types of patterns throughout all of the 16,000, I guess, nano chromosomes. And it's clear how the scrambling can happen if you have a molecule that, well, it turns around like this, right? Then there's exchanges in this part. And we end up with a molecule of this sort, which is the repeat word. Or if I have just folding of the molecule and exchange, and we have a return word pattern. So these are these are explanations for these 2 here. Why are they so most prevalent? And I think Laura has also shown that evolutionary, through the evolutionary process, the ciliates prefer to add extra vertices in this either repeat or return segments. That means that the scrambling happens in this form or in this form throughout the evolution as a preferred way to do it. What happens to the embedding of these things in the in space? Well, these are loop insertions, so the genus doesn't change. The genus of the graph doesn't change at all, and the return pattern also doesn't change the genus because this is a loop and this is nested loop addition to my graph. So I don't have any changes in the genus range. On the other side, this thing, which is most prevalent, if you remember my 3 vertex graphs that showed up in the data, this was the most prevalent one. Turned out that it can show the genus range depending whether we have even or odd addition, even number of vertices or odd number of vertices addition. It can change by adding 1 or 2 to the genus range, or also in the case of odd, we can subtract 1. So it fluctuates up and down. With the tangle chord, those really complex parts, actually, they have very narrow genus range. The tangle chord has genus range n-1n if I have 2n-1 vertices or 2n vertices. In both of these cases, this is my genus range. So there are only 2 genera that of the surfaces that this tangle chord can be embedded at. So it's either n-1 or n. And if I add this to my graph, then I'm actually adding this type of genus, this type of interval in my genus range. So it becomes a bit more complex. Okay, I'm just about to finish. These odd, even, repeat, and return patterns actually turned out to be single appearance was about 81.7% of the data. And if I look at the nesting appearances of this, one inside the other, then we can explain about 96.4 of the scrambled genes. It will be interesting to see whether similar patterns appear in other types of rearrangement processes. If I have patterns, then maybe rearrangement is not random. Maybe these patterns have some better selection, adhere for better selection processes. I don't know, could be. My last remark is that there are very nice mathematical problems that can arise during working in biology and trying to understand biological processes. So we need to be more involved. And this is my group. I have, Jonathan Burns have worked on this, and Mustafa Hadjic, and Jasper Brown has been working currently on the data that is new sequencing that Laura is producing. Laura and her lab, of course, is essential in everything that I said today. And thank you very much.
Thank you.
Okay.
Thank you. Questions?
Thank you.
Any questions? Somebody doesn't jump on a question, then I will.
Okay.
So
this is not what you probably expected me to talk about, right?
oh, no, this is cool.
I was debating myself, should I talk about topological data analysis and how that can show up here, and the recombination, and looking at the homology, describing this. And I said, I have never talked about the genus range, so I decided to talk about something I have never talked before.
Yeah, no, that's nice. This is cool. So I assume that these are not diploids, right? That when the recombination takes place, that there's just a or am I wrong? Are these diploid organisms?
The micronucleus is a diploid. The macronucleus is multiploid, and as I mentioned, I think Laura, I don't know if you're still here, but is that true that the copies of the nanochromosomes in the MAC appear in hundreds of copies? Maybe she disappeared. I don't see her.
So, okay, so the next question I was going to ask was how stable the patterns are for a given config, right? I mean, you can imagine that so maybe the diploid. Okay, so you have your config, right, which is a small little chromosome, right? And then there can be mutations that take place on the chromosomes that could, in particular, could give you different of these, looping, returns, repeat.
Right.
Do you know anything about how that's distributed? I mean, is this really very, very tightly bound on a particular config, or is there a wide range over a config?
Actually, the situation is much more complicated than I described. This is just very simplified version of it. We had a recent paper where we see that it's not tightly, one gene can interact with another gene. So there may be interactions between different contexts on the micronucleus. Okay. So I may have, this is where the topological data analysis was coming from, but you can have two different contexts and the segments of one context to be entirely inside the segments of the other one, in between the segments of the other one, either like this, or flipped around. So there is quite a bit of interaction. So this is just considering one nanochromosome and one type of molecule. Mm-hmm. If I look at the genome complexity, then I think there is way more complicated stuff than I just described. This is a simplified version.
Yeah.
And
okay.
I don't know how the distribution of this thing actually is. The problem with the distribution is, well, Jasper is currently writing a program that is doing the annotation for the new sequencing. The annotation is many to many. I may have a segment in the micronucleus that can be used in this gene and also in that gene. And I can have two segments that are used in the same gene here. And this many to many association between the segments is, it's becoming much more messy.
Because I was, one of the ways that, again, I always hesitate to pretend like I know any biology, but I mean, gene duplication is one of the ways that evolution acts, right?
Right.
And here somehow because we have these little segments, the little segments can duplicate as long as they keep the right pointers, right?
Well, the segments don't duplicate themselves. I think that you duplicate the whole chromosome and the whole thing. The chromosome contains the segments in certain order, and they're all duplicated in that particular order.
Well, that's for replication, right? But I'm just talking about mutations, right?
So, the mutation, okay.
Right. So because you were talking about how do these things appear, right? So for me, this is
a
dynamic process, right?
Exactly. That was this part here.
Right. So, I mean, this should be a dynamic process that generates this over time, right? And so I'm just trying to think of, what kind of has to be preserved and what can be changed. Obviously, you can get base pair changes, right? But yes. One of the big evolutionary, one of the big things in evolution, making evolutionary changes, is gene duplication.
Yes.
And here somehow the pointers are really important because because you're not really looking at the whole gene, you're looking at a fraction of it that's then going to get reassembled, right? And so you now have the ability to kind of duplicate something that when it goes to the macro gene, it fits or doesn't fit, but you now have room for lots of variation within there. And I just wonder, yeah, I don't know. I was just wondering if things like that could be used to explain the
Yeah, well, it could be. I haven't thought of it. It's an actually interesting observation, because you know that the pointers are not very big.
Mhm.
There's short pointers, in particular for these loops, the pointers are pretty short, 2 to 3 nucleotides, so that they're not that interesting. But for these guys that are more complex, you can have up to 50 nucleotides. But certainly on average about 20 nucleotides. So there, you know, repetition of 20 nucleotides is not that frequent. On the other side, the genome itself is about 60% ATs and 40% or maybe even a little bit less Gs and Cs. So if I look at long sequences of As and Ts, you know,
Yeah.
it may happen very easily.
Yeah.
I want to ask a short question if I can. So there are systems where you have alternative splicing of genes in other organisms. Is this a similar? And so is it like the same way that the pointers are now repeated for different MDS? You have the same pointers and then you can just put things in different order, in particular in the humans, and do immune system, I think, is being generated semi-randomly. So
right. So alternative splicing happens on the RNA level, right?
Okay.
And it usually you have different exons combined to produce the different genes. Here it's very similar to that, but one step away. Here actually I have the DNA first scrambled. So I have to process the DNA. And then once I get to this processing to DNA, then I have the next step is the alternative splicing with the RNA.
Okay.
But the process itself is pretty similar. That you shuffle the exons. I mean, in alternative splicing, you shuffle the exons to get the new RNA.
Okay. Thank you.
But it's interesting that this is happening at the DNA level, one step before it gets there.
Mm-hmm.
Are there other questions or comments? All right, then let's thank Nataša again. And yes, I like what you talked about.
Okay.
It's cool.
It's probably different than you expected, right?
It is, but it's cool. It's cool.
All right.
All right. So yeah, thanks, and we'll meet again in 2 weeks.
Yeah, thanks again. That was great.
Hopefully we
you.
meet in person at some point.
Yes.
Exactly.
The invitation to come up to Rutgers still stands.
All right.
Someday when we can travel again. All right. Take care.
Bye-bye.